Hello, everyone. My name is Kyla Morris, and today I will be presenting to you the identification of cellulose-utilizing bacterial species from the rumen of beef cows. Just a quick introduction. Angus cows are beef cows that mostly eat grass, hay, and silage. They are ruminants, and their digestive systems consist of four stomachs. These four stomachs include the rumen, the omasum, the abomasum, and the reticulum. Cows have a symbiotic relationship with the bacteria that reside in their rumen, and they help digest the feed that they eat. Different bacteria in the rumen are used to break down different polysaccharides. So some bacteria specialize in breaking down starches, while other bacteria specialize in breaking down cellulose and so forth. Bioreactors were used to keep the bacteria in the cultures alive. PCR and qPCR was used to make copies of the DNA sequences within a short amount of time. Gel electrophoresis is a technique used to separate charged molecules according to their charge and size. So DNA, RNA, and proteins are separated with gel electrophoresis. And then bioinformatics is helpful in identifying different operational taxonomic units or OTUs and their abundances. Many of the bacteria that reside in the rumen are unknown bacteria. The objective of this experiment was to identify the most abundant of the different bacterial species within the rumen that digest cellulose. The materials and methods of this experiment consist of the following. The first thing I did was obtain rumen fluid from cannulated cows on the South Dakota State University cow-calf unit farm. Once the rumen fluid was collected, it was brought back to the lab within an hour or so, and the cultures were prepared. These cultures were kept in bioreactors or bottles that were stored in incubators. And these cultures were enriched with cellulose and bactotrypto. Bactotrypto is like a protein for bacteria. Each day from day 0 to day 14, we obtained the pH of each culture to ensure that there was an acidity increase or that the environment itself was acidic. This indicated that bacteria was growing. Next, we extracted the DNA from rumen fluid samples, and this pure DNA was used for PCR and qPCR analysis. The results from the PCR analysis were loaded into the wells of gel for gel electrophoresis. We performed bioinformatics. I was provided with custom Perl scripts to be able to do this. Once we were able to cluster all of the operational taxonomic units, we were able to put these files into RDP classifier, which gave us the taxonomic names and abundances for all of the bacteria in our cultures. We were able to analyze this data with bar graphs and histograms. In this picture, there is me and a cannulated cow. The cannula gives us direct access to the rumen without having to put the animal to sleep or anything like that. So you simply remove the rubber circle within the middle of the cannula, and there is the contents of the rumen. So there is like half digested grass, hay, and silage in there, along with the rumen fluid. So we manually stuck our hands in there, pulled out the contents, and squeezed it so that we were able to get majority rumen fluid. We used about 3 cows for this. Here's a picture of the bioreactor. The bioreactor has a orange blanket around it. This orange blanket is called a thermal blanket, and it's set to 38 degrees Celsius, and that's because that is the temperature inside of the cow, which is the optimal temperature for these bacteria to grow. At the top, you can see that there is collected grass and a little bit of molding. Within the middle of the bioreactor, there is a rod that spins at 150 rotations per minute to basically stop the grass from collecting at the bottom of the bioreactor. And then there is a tube at the top of the bioreactor for all of the gases that are coming from the bacteria doing their job basically and fermenting so the gas can have a way to escape. Here are my results for qPCR. Two primers that specifically gave us bacteria that were amplified in our samples was BT12 and BT19. As you can see, the CT values are really high for the control or no DNA, but in the other columns, you can see that they are lowered. The lower the CT value is, the more DNA that is present within the culture. For this slide, you can see that there is the initial bacterial phyla. So the most abundant phyla that were initially found in the rumen were Bacteroidetes and Firmicutes. In these graphs, you can see that Gamma proteobacteria, Synergistia, unclassified bacteria, and Firmicutes were the most abundant to grow in our cultures. Gamma proteobacteria is the most abundant in the day 7 cultures. And then if you look to the left of the blue columns on the first chart, you can see that the orange that represents the Synergistia begins to expand. So I would say that towards day 14, the gamma proteobacteria began to die off and the Synergistia began to amplify even more. In the graph next to it, you can see that unclassified bacteria had the most abundance in the day 14 cultures of cellulose A and cellulose B, but in the day 14 culture for cellulose C, Firmicutes seemed to be the most abundant. So my conclusions for this experiment: the bacteria in the rumen that are responsible for digesting cellulose belong to one of the 4 phyla: Firmicutes, gamma proteobacteria, Synergistia, and unclassified bacteria. Samples day 14, cellulose A and cellulose B show that most of the bacteria that survived off of cellulose were unclassified bacteria. Sample day 14, cellulose C showed that bacteria belonging to the phylum Firmicutes survived the most. Gamma proteobacteria seemed to be the most abundant bacteria across the cultures on day 7 and then seemed to rapidly die off while it approached day 14. Here are my references. Thank you for listening to my presentation.